Hello. In this video, I'm going to provide a solution for the homework of the workbook on page number 36. What you have to do, you take this workbook and watch this video and try to learn each and everything. After that, you have model answer at the end. So you copy this model answer to the space. This is 6 marks question. So let's get the background for this question. This is the pancreas. Pancreas has two functions. Number one, exocrine function, the meaning it secretes enzymes for digestions, amylase, trypsin, and lipase-like enzymes, and some other enzymes. The second function is endocrine functions. Endocrine functions, it secretes hormones such as insulin and some other hormones like glucagon. So here we focus on insulin. When insulin is secreted, the insulin can directly enter into the bloodstream. So when insulin goes to the blood, insulin can distribute all over body. And what is the function of insulin? Insulin lower blood glucose level. So insulin helps to reduce blood glucose level. So after a meal, carbohydrate increase glucose level in the blood, and then this glucose level again decrease in the blood. So how insulin lower the blood glucose level? So I'm going to show you like this way. This is a cell and it has a phospholipid bilayer, and a glucose molecule cannot enter to the cell because this phospholipid bilayer prevent entering glucose into the cell because it doesn't favor the molecules like a polar large molecule to pass through this lipid bilayer. In that case, this is the insulin. Insulin has a receptor on the cell membrane of the cell, which is very much complementary. That means only insulin can bind to the receptor. So here, insulin bind to the receptor. So after insulin bind to the receptor, so I play this again as it just, okay. So here, insulin bind to the receptor. After insulin bind to the receptor, then there is a cascade of event. That means many molecules, enzymes, they activate in the cytoplasm. So I can show this as an arrow. So this finally lead moving the glucose transporter to the cell membrane, allowing glucose molecule to move through this transporter into the cell. So insulin stimulates some of the enzymes and chemicals to translocate the transporter to the cell membrane, and glucose can come in. Another function of insulin is it can convert glucose into glycogen. So here's a glucose molecule. Enzymatically, it converts glucose into glycogen. So enzymes like glycogen synthase is stimulated by insulin. So insulin take glucose from the blood into the liver and store as a glycogen. The type 2 diabetes, 90% is common, attributed to the lifestyle changes, and people have different lifestyle. With the age, type 2 diabetes increasing. So there's another one, type 1 diabetes, which is the cause is very different to type 2 diabetes. And we only look the type 2 diabetes. And some of the thing about type 2 diabetes, 540 million people in the world affected with diabetes type 2. And the alarming thing is, all over the world, the diabetes population increasing. So here in Africa, 134% increase in next coming up years, 50% to South America, and even Asia, Middle East, these countries, it's going up in higher rate. So it's a very important thing to consider, the type 2 diabetes and treatment for type 2 diabetes. And another important thing is the large number of people die in the world that come as number one in the world now, the cardiovascular disease. So diabetes can increase cardiovascular disease. People who have diabetes, type 2 diabetes, they are risk of getting cardiovascular disease. So risk for cardiovascular disease is high when person has diabetes mellitus. Okay, the background is over for this question. That knowledge is very useful for you, all your biology and coming up questions. Type 2 diabetes is a disorder that can result from ineffective or insufficient insulin. So this happens if the insulin level is down, or as you see in this previous slide, there is a receptor and insulin can bind to this receptor. If there's a problem of receptors, not enough receptors, insulin resistant to the receptors or anything, or this one, insulin decrease, can increase getting diabetes and then cardiovascular disease. In this experiment, scientists use cinnamon, and already there is evidence that cinnamon can increase uptake of glucose from blood into the liver, increase synthesis of glycogen. By doing that, it can lower the glucose level. In a study using rats, the effects of cinnamon extract on level of glucose in the blood was determined. In this study, group of healthy rats and group of rats were induced diabetes were used. The rats were given either the cinnamon extract or water each day for 30 days. Water is used as a control. The table shows the result of this experiment. The times 0 to 30 days, rats given water, rats given cinnamon extract, and here rats given water, rats given cinnamon extract. And this is the healthy rats, this is the diabetes rats. So here we can separate these two. This is healthy, this is type 2 diabetes. And both group, water and cinnamon extract, water and cinnamon extract. Water is a control, cinnamon extract is an experiment group. Now, the question is, explain the result of this study, 6 marks. So, when you have to do explain the result, the format is DEC. Describe, and this cover the explanation, that is how it happened, and this cover any comparison between groups. So it include the description, explanation, and compare and contrast part of the answers. So explain the result of this study. First of all, you need to make the format for this question. So only one table. You had used this table for explanation of result. I think this is quite easy for you now. If you have one, so consider this is the one unit of result. So we are going to fragmentation now, the fragmentation process. In fragmentation process, I'm going to separate this as a one result, the second one as a one result, third one as another result, and fourth one as another result. And further, if you look this uh, these four groups, three groups. So these three groups mostly used as a control because the water is a control, and here water is a control, and here healthy rats cinnamon extract is a control. So this part is the most important one for the experiment. Now what you do, you just describe this result and give the explanation. Describe the result, give the explanation. Describe the result, give the explanation. Describe the result, give the explanation. And comparison, we can do comparison like this. So here the values are really high. And here is again values are higher. So that means the glucose level is high as they have a diabetes. So whenever we can compare like these values with the rats given with water here and rats given with water here. So generally the diabetes group treated with water, it's higher. That's a kind of a comparison. So now we do like this. So we take the first part and rats given with water, what we can notice, the 0 days 160, 106, sorry, and then 30 days 94. So it's like a 12 whatever milligram per dm cube decrease. Now how we can justify this and what we say about this decrease? Is it a very important decrease? So it's a significant level of decrease, and anything we want to make some kind of comparison between these 0, 10, 20, 30 like days, we have to look. So we can say compared to the values which you have seen in the other groups, for example, like 300, 400 levels. So there is a small change, decrease. There's a little decrease. The description is there is a small decrease. There is a slight decrease of the glucose level in healthy rats given water. This is obvious because these rats, they do not have water, and healthy rats, they can control their glucose levels as they have an insulin. So that one you can write now. Healthy rats that were provided with water experienced a slight decrease of regulating normal level. This is due to the production of insulin by the healthy rats. As a result, the healthy rats were able to naturally regulate their blood glucose level. So I try to use this as a description, and that is explanation. So that group is finished, and we are going to the next group. Rats given cinnamon extract. Again, there is a decrease 12, let's say 2, 9, 10, 12, similar to the group of rats with water. There's not much different in the comparison. And we can say again, there's a slight decrease. This slight decrease is due to maybe some effect of the cinnamon extract. They also have a normal insulin level, so they can regulate the blood glucose level. Healthy rats that were given cinnamon extract, a very slight decrease in glucose level. This has been seen in rats given with water. So here again, I use a comparison. the two groups, water and cinnamon extract. So this is the comparison. The results are comparable. Alternatively, the glucose level in healthy rats remained very similar even after being given cinnamon, indicating that cinnamon had no additional effect on rats. So give it a third group now. So third group, the rats, they have a diabetes. As they have a diabetes, the glucose level is always higher, even the starting day 395. And when this group is given with water, this again increase. If you look at the last number here, this one is increased. Blood glucose level increase when there is no treatment, because water is a control. So we can write that the blood glucose level increase as they have a diabetes that they can't regulate the glucose level. Diabetes rats that were provided with water experience an increase in glucose level surpassing those of healthy rats. The occurred due to the absence of insulin in diabetes rats, which is responsible for regulating blood glucose levels. So their glucose level increase. This is due to they have a diabetes and insulin is no longer there to control. Now we are coming to the last group. Starting glucose level is 378 and the end glucose level is 369. So let's see each value now, as this is the most important group, treatment group. The diabetic rats are treated with cinnamon extract. The starting number 378. So this decrease here by 346. Then again here increase. Then again here decrease. But overall, if you take 30 days trial, 78 to 369. We cannot say there is a decrease because the final value of 369 is just like a 9 milligram per dm cube decrease. As we see in here, 12 decrease and here also 12 decrease. So comparing that, we can't say 9 is a decrease, but we can say with these two groups, insulin level remain constant. So they regulate insulin. So this kind of increase and decrease, we use the word fluctuate. Fluctuate and regulate. So insulin level fluctuate with the measured days, but it's regulate. So it doesn't increase compared to this. So here it increase, but here it's regulate. Now you can write that rats with diabetes administered cinnamon maintained relatively stable glucose level, which were higher than those of healthy rats. Glucose level in unhealthy rats fluctuated depending on amount of glucose utilized in this process. The explanation, cinnamon facilitated the transfer of blood glucose to the liver, where it was converted into glycogen by enzymes, preventing it from re-entering the bloodstream. The effectiveness of cinnamon in regulating glucose level was found to be lower compared to insulin, possibly due to poor absorption, slow action, or limited enzyme stimulation. It has an effect, but it is not that great effect, but still it can regulate glucose level. This is due to it can still do certain level of enzyme activations, absorb, and then they can convert to glycogen, but this effect is not that great as insulin. So now we coming to the end of the question. So all these things, the answer, that's a model answer. I took it to the one place now to the screen, and you can copy this to the page on these questions. So please do this because it's very important. So eventually we want to target the higher mark. So you have to do like this until you are get 100% confidence in writing answers in upcoming exams.